We'll wait if it does. Okay, looks like she's still there. So propagation, there are two types. There is continuous propagation, which is kind of like it sounds. You know, the impulse starts at point A and it spreads gradually through to point B, then to point C, then point T. Okay, these are propagations, action potentials that occur in unmyelinated axons. So to remind you of your axon, the myelin are those little kind of sheaths around the axon. So they looked like little bumps, right? And there were breaks, okay? Saltatory propagation is kind of like the jumps, okay? It spikes in the internodes because the ions can't cross the myelin. Now this is beneficial because it's faster. Instead of continuously going A, B, C, D, it goes A, then jumps to C, and then jumps to E. And so it's a quicker propagation. And there are pictures on page 422 and 423 of continuous and saltatory propagation. And it's just trying to demonstrate how, you know, continuous is, you know, along those same lines, whereas saltatory, and you have a myelin sheath, so it has to jump. And so it's faster. Synapses. So we do have to kind of scooch through this last section. Synapses, of course, are where a presynaptic and a postsynaptic neuron are going to be not necessarily touching, but very close. Okay, so this is where information is transferred from a presynaptic to a postsynaptic. There are two types, chemical and electrical. And chemical, I'm going to go through very quickly because we've talked about chemical before. We talked about acetylcholine with the neuromuscular junction. All right, so this is most abundant, and it relies on a release of a neurotransmitter, and the neurotransmitter passes through the gap and then triggers some response in the postsynaptic neuron. If it does release acetylcholine, it does have a fancy name, cholinergic synapses, although we most often talk about acetylcholine. And so these are just the events which we have talked about before, so I'm going to skip through that today. All right, synaptic fatigue. So before we talk about electrical impact in synapses, we want to talk about fatigue and delay, okay? Because the synapse is not a continuous machine. A continuous machine. Instead, there are some things that can like cause it. The synaptic fatigue is when a synapse stops functioning, okay? The neurotransmitters are then reabsorbed and recycled. And so it's all due to the neurotransmitter. So the synapse cannot function without the neurotransmitter, so that needs to be replenished. So that's kind of the idea of fatigue. A delay is just what it sounds like, the lag between the action potential at the terminal and the effect on the postsynaptic. So the time it takes the neurotransmitter to actually leave the presynaptic neuron and affect the postsynaptic neuron. Okay, electrical synapses we have not talked about. They are not common. However, they are found all in the brain and spinal cord. This is a synapse that is joined by a gap junction. So this is one of those cellular junctions. A gap junction actually, and you can kind of see it down here, connects one membrane to an adjacent membrane. And there's a channel. So the axon terminal here of the presynaptic neuron is connected to the postsynaptic neuron and the dendrites of the postsynaptic neuron. So this has to rely on changes in membrane potential and not a chemical. So the impulse is going to travel down, and that's what these little red lines are, travel down to the synapse, And that change in that membrane potential of one cell, because they're connected physically, will affect the membrane potential of the adjacent cell. All right, it's quicker, it's much quicker, because you don't have to wait for the neurotransmitter to cross the synapse. This is just almost immediate, but it's just not as common. All right, questions so far? Stop me if there's questions, or if I've skipped past something you want to go through again, just stop me, because we are 20 minutes from class. We're on page 426. Postsynaptic potentials. There are two types. There's an excitatory and an inhibitory. So let's talk about those two types. And these are graded potentials. So, you know, they're dealing with the membrane and a change in the membrane potential. Okay, excitatory. So let's talk about excitatory first. If you look on page 426, we're going to talk about the bottom graph there as we describe this. So this is a graded depolarization. Remember, depolarization is when you have a change in the membrane potential, it actually rises, okay, becomes more positive. It's close to excuse me, it's closer to threshold. So that threshold, remember, with the action potential, is that all or nothing point where the impulse is propagated. If it doesn't reach that threshold, then the action potential kind of stays local. It's called facilitation when the membrane is it's not what am I trying to say? More facilitation means less stimulus is needed. If it's facilitated, it means it doesn't need as many of the impulses from the stimulus to continue depolarization. So excitatory, think depolarization. Inhibitory, it means that you need a more of a stimulus. So as opposed to less of the stimulus with the excitatory, the inhibitory, you need more of a stimulus. And so this results in a graded hyperpolarization. Hyperpolarization meaning it's becoming more negative. So we're talking about postsynaptically two types of potentials that helps process information and relay information in a neuron. All right, so then we talk about summation, which is looking at both together. The collective effects of both excitatory and inhibitory postsynaptic potentials. So the arrival of two different neurotransmitters at the same time opens two different sets of ion channels. However, the net effects may be no change in membrane potential. So it is the sum of all of the effects that's happening at the synapse. Summation here, and this is the example, if you've got a hyperpolarization stimulus and a depolarization stimulus, aka the two different neurotransmitters, you can see that they negate each other out. So your membrane potential stays the same. If you look at the bottom of 426, you will see how this might be possible. You will see a, like the main thing is the axon there, that's kind of the purple background. And then you see all of these dendrites, which are purple. But then you see all these little yellow and blue parts, okay? The blue parts are the axon terminals. That's what we're talking about. There are multiple synapses in one axon or one dendrite. So that's where you can see, okay, summation is actually taking all of the effects of all of these different axon terminals and seeing what the net effect is. Does that make sense? Okay, so and that's the picture. I guess I have that on there. And that's what I was just trying to describe is a single neuron may receive information from thousands of synapses. There are two types of summation, temporal and spatial. So let's talk about those. Temporal, when there is a single synapse stimulated repeatedly. So if you look down at the picture, you can see first just one axon terminal and a barrage of stimulus. And that propagates the action potential. The other kind is spatial. And if you think about spatial meaning, you know, a physical distance, this includes two or more stimuli at a similar location, and there's multiple synapses, multiple stimuli, and that propagates the action potential. So in the body, there are neurons everywhere, and they look different. You know, they have we're just talking about like all the possibilities of neural processing and neural relay that are in the body. All right. Let's flip the page to my favorite page, 428, which has a whole bunch of neurotransmitters on there, okay? And we're talking about regulation. So regulatory neurons are neurons that facilitate or inhibit activities of presynaptic neurons. So it's actually like, if you think about a presynaptic and a postsynaptic, it's actually like a presynaptic. Okay, it's before the presynaptic, and it's going to regulate whether the neuron is processing information or not. And that's what these green guys are. Information processing also includes the neurotransmitter. So if you just kind of take a look at 428 for a second here, and take a look at some of the words, some of the neurotransmitters, you might actually be familiar with some of them. Hopefully you're familiar with acetylcholine, since I've said that a million times. You've probably read it a million times and seen it a million times. But then there's norepinephrine, epinephrine, serotonin, and you might not have heard of glutamate or gamma aminobutyric acid, which is GABA. There are 100 different neurotransmitters or so, 100 or so. These are the ones that are common, that are most studied when you look at an anatomy class. Acetylcholine is most common, okay? It's in the neuromuscular, it's in the central nervous system as well. Norepinephrine and epinephrine, if you think about it, epinephrine is the same thing as adrenaline. So epinephrine, if you look at the comments there, it has excitatory effects. So if you think about anything that will cause your adrenaline to get moving, epinephrine is also going to get moving, because epinephrine and adrenaline are interchangeable terms. Epinephrine is also a hormone that affects other areas of the body, but in this case, it's a neurotransmitter. Information processing also includes interpretation, okay? So far we talked about regulatory neurons, neurotransmitters, and now interpretation, all right? Interpretation relies on the frequency of the action potentials, all right? For example, muscle contraction, which is the response, the end of your neural information, neural relay, that is going to depend on the frequency of action potentials. And if those action potentials stop, then your contraction is going to stop. Makes sense? Oh, and that is the end of chapter 12, is it? Yes, it is. Awesome. All right, so we do have time. Take a couple minutes here. When I said action potential, Are you thinking of like the depolarization, repolarization cycle? Like what are you thinking of, and what can I revisit with this extra time? What do you want to talk about with action potentials? Because it's really, really important that you understand an action potential. If you flip to 413, that's the start of that section. It's all about action potentials and membrane potentials. You can look at some of the pictures. I'll just give you like a minute or two. If you want to go over anything else with action potential or go over anything again, now is the time. The steps are the steps are on 420. So everything up to that is kind of explaining the structure. Let me go ahead and stop recording and then